All right. Hello, everybody. This is a recording of the second lecture for Chapter 10, mostly about muscle physiology. We will pick up the pace where we left off after the first lecture. And on Monday, when you come to class, we will conclude this Chapter 10 with a highly entertaining third lecture. So let me go to the screen share and we'll get going with the slides. I have a number of animations and I hope you find it super entertaining to learn about what starts and ends muscle contraction, how this is all regulated, and also where muscles get their energy from. So let me go to the screen share. And let's make this full screen. So this is where we left off last time. This square somewhere else. So this is where we left off at the end of the first lecture. We looked at the elements necessary for muscle contraction. We analyzed all and named parts of the sarcomere. And this was the snapshot we ended up with from going from a relaxed muscle with relatively long sarcomere to a shorter and an even shorter sarcomere in a fully contracted muscle. This shortening of the sarcomeres create a force. On the right here, you see the electron microscopic view, and this is why solving the puzzle of how muscles contract was taking so long of a time, because looking at these images, scientists just couldn't figure out what exactly is happening here. But after the Second World War, this all became clear. Individual proteins were analyzed, and so this sliding filament model is how skeletal and cardiac muscles contract. The sliding filament, of course, means that we have the thin filaments that are being dragged along the thick filament towards the middle of the sarcomere. So the thin filament is sliding over the thick filament. And I'm not going to say all this. This is just a summary of the last portion of the first lecture. So once we have the players to enable skeletal muscles to contract, the question is, how can we instruct the muscles to start contraction? The natural state of muscle cells is a relaxed state. If you want to make them create force to create any kind of movement, and in the lab, you all have seen a number of muscles, whether changing facial expression, elevating the mandible, depressing the hyoid bone, elevating the scapula. Muscles have to create force to carry out all those actions, so contraction must start. It is our nervous system that's in charge of skeletal muscle contraction. So for skeletal muscles, we can decide at will. Some skeletal muscle movements are automated, but here we are talking about the scenario, just what are the elements that our nervous system and skeletal muscles need for this information transfer. So the information from the nervous system is called a nerve impulse. This is coming down to the end of the axon, and we'll have a part of the axon release a chemical, and this chemical, acetylcholine, is going to instruct the muscle cell to open sodium channels, and sodium ions will rush into the cell. We will be associating this sodium flow into the muscle cell with muscle excitement, muscle action potential. So a flow of sodium at this stage will be instrumental to tell the muscle cells it's time to contract. So we're going to look at these two steps in great details. Obviously, we're going to look at parts of this neuromuscular junction, because this is where all this action happens. On this slide, you are seeing an a scanning electron microscope view of the large log-like skeletal muscle cells. And then you see structures like roots of trees surrounding these muscle cells, in many cases, as it's shown here, contacting the skeletal muscle cells. So this contact area is what we call the neuromuscular junction, or NMJ. And the ends of the axon is the axon terminal. On the surface of the skeletal muscle cells, there are these depressions. These are called the motor end plate. And at the end of the synapses, there are these swollen areas. We're going to have an artistic rendition. I think we'll be a lot better than this. But it's good to see how a real living neuromuscular junction looks like. So the axons from our nervous system make contact with skeletal muscle cells at this particular location. So here is the artistic rendition of this, and in a few minutes we'll go to an animation. So major parts here, where the axon comes to an end, it widens out. This is called the synaptic end bulb, because it's like a bulb of a plant. Inside the synaptic end bulb, there are these vesicles. Now, way back in chapter 3, we mentioned secretory vesicles that are tools for the cells to shove material out of the cell. And this is what's going to play out here. These synaptic vesicles are secretory vesicles loaded with the chemical acetylcholine. Acetylcholine is a small organic chemical. It's commonly used for information transfer. We'll be seeing that in chapter 12 in the nervous system too. So when a nerve impulse wanting to tell the muscle to contract comes down to the end of the axon, this information needs to be transferred to the muscle cell. On this little smaller magnification, you see the large muscle end plate, this depression on the surface of the skeletal muscle cell. And here in this more magnified view, you are seeing the burst of the secretory vesicles as they release their content. In chapter 3, we would call this exocytosis, right? The release of the content of secretory vesicles. And this happens here with every nerve impulse coming down here. Secretory vesicles are pushed to the edge of the axon of the synaptic end bulb, and the content of the vesicles are dumped into this gap here. This is called a synaptic cleft. What happens here is that this acetylcholine will have detectors. We call them receptors. So when you see this term ACH receptor, it means acetylcholine sensors, detectors, these bluish structures here. And once acetylcholine sticks to the receptor on the muscle surface, the receptor opens up and opens a channel. Now, through this channel, sodium ions, these green bulbs, are going to stream into the cell, and flow of sodium ions into muscle cells means muscle action potential, muscle excitement. So through this process, we excited the muscle cell, making it ready to contract. We successfully moved the transformation from the axon to the muscle cell, and the instruction is start contracting. So this is the first step in this multi-step process, getting the muscle cells excited by allowing sodium ions to rush into the muscle cell. When there's no acetylcholine around, the channels are closed. It takes the acetylcholine to open the door for these channels and allow sodium to rush through. So let's take a look at an animated version of this. Let me see if I can go to another part here. And let's see. I think this will be great in this magnified version. So what we see here is the muscle cell with the motor end plate, this depression. You are seeing the acetylcholine receptors here. Here is the axon end bulb. We have the synaptic vesicles or secretory vesicles loaded with acetylcholine. And at this time, the muscle is still relaxed. Sodium ions are here in the synaptic cleft. So let's start this animation. And here we see the action potential coming down to the end. And after this calcium flow into the synaptic end bulb, take a look at what happens to the synaptic vesicles. Acetylcholine is dumped into the synaptic cleft. And then acetylcholine will stick to the acetylcholine receptor and opens up sodium channels. I'm going to stop this animation right here for now. Take a look at the door open once acetylcholine binds to this receptor. Let's run this animation a little further. And now you see that the sodium ions will be rushing through into the muscle. And so this move of sodium ions into the muscle cell creates the muscle excitement, the muscle action potential. So at this point, again, we are successfully excited the muscle cell. The flow of sodium ions into the muscle cell is a chemical signal that will be picked up by other parts of this muscle cell. And eventually, the information will be transferred to the sarcomere where contraction will begin. So let's stop at this point for a minute. We have a few other things to say about the synaptic neuromuscular region. So there are a number of disorders that can affect the neuromuscular junction. Botulinum toxin is a bacteriotoxin from the Clostridium botulinum bacterial species. And this is a bacterium that colonizes food. So food poisoning, most of the time, is a mild form of this botulinum poisoning. In case of heavy botulinum poisoning, the toxin from the bacteria will block the action of the neuromuscular junction. Botulinum toxin stops the release of acetylcholine. When there's no acetylcholine, the muscles will not get the signal to contract. Muscles remain relaxed. The deadly aspect of botulinum poisoning is that it can cause death because the diaphragm is a skeletal muscle and it can be paralyzed. When our diaphragm remains relaxed, we have no inhalation, and that leads to respiratory stress and eventually to death. Botulinum toxin, if you take the first few letters of these two words, this ends up as Botox. So a purified form of this toxin can be injected into muscles, and this Botox treatment makes the muscles relax. Think about from a lab, the action of frontalis muscle. If somebody wants a smooth looking forehead for cosmetic purposes, Botox treatment can block the action of that frontalis muscle, and the skin of the forehead will not be wrinkled by contracting frontalis muscle. Here's another agent, curare. This is made by plants, especially South and Central America. Native Americans in this part of the Americas dipped their arrows into this plant secretion, and when they hit any kind of animal with this arrow, the animal got paralyzed. So curare, as it turns out, also paralyzes muscles, force them to relax. Curare acts a little differently. It blocks the acetylcholine receptors. Think about the curare maybe as an umbrella over the acetylcholine receptor. It stops the acetylcholine from contacting the receptors. It has the effect of muscles not getting the signal that they need to contract, and muscles remain relaxed in case of curare poisoning. There's also an autoimmune disorder where our own immune system attacks and destroys the acetylcholine receptors at the neuromuscular junction. The more receptors are destroyed, the weaker the muscle gets. Myasthenia gravis, as this disorder is called, is a progressive disease, and at the end stage of myasthenia gravis, again, the disablement of the diaphragm is that causes a life-threatening condition. So what happens after this nerve signal is passed onto the muscle cell? So step one here, there's a curious statement: muscle cells become more positive. This means to say that inside the muscle cells, there's an accumulation of positive charges. Now, you saw the sodium ions rushing into the muscle cell, and those positively charged sodium ions are responsible for this. So when we open the acetylcholine receptor sodium channels, the muscle cells become positive because of that rush of positively charged sodium ion. This action will trigger the opening of other sodium channels throughout the muscle membrane and also throughout the transverse tubules. So sodium ions will be flooding into this muscle cell in the middle and on the sides of this muscle cell. Wherever we have cell membrane, sodium ions will be rushing in to the muscle cell. This positive charge will force the SR, sarcoplasmic reticulum, to release calcium. So this is the second step. Another ion is playing the role here, calcium ion. And as a third part, calcium ion now floods into the sarcomeres and changing the shape of troponin and allows myosin to contact actin molecules, and contraction will begin. So let's take a look at this in a more detailed fashion. This is your textbook's illustration. We have an animation, I think, that animation or two that will be very useful. But here's this process. So this is not illustrated super well, but take a look at the relaxed muscle. The myosin head groups are not contacting any of the thin filaments here. This is a real essential part. If you want myosin head groups to contact the thin filaments and begin contraction, we have to go through this step. So we need to have the muscle action potential, the flow of sodium ions into the cell. That will release calcium from the sarcoplasmic reticulum. And you see these purple calcium ions flooding into the sarcomere. And the purple calcium ions is barely seen here, binds to troponin, and troponin will change how the thin filament looks and now opens up the contact for myosin molecules, and contraction will begin. So before we go to the contraction cycle, let's take a look at another animation. I think will be a little better illustrating this. So Let's go here. So on this animation, we are seeing the muscle cell membrane. The action potential means the flow of sodium ions rushing into the cell. You will see this action potential go through the T tubules. These large yellow bags are the sarcoplasmic reticulum, and we will see them release calcium, and those calcium molecules will cause the change here in the sarcomere to begin contraction. So I'm going to stop this periodically, but let's start with the action potential, and let's see how calcium is released from the sarcoplasmic reticulum. So here you see the action potential going all the way down the T tubules, allowing sodium ions to rush into the muscle cell. And now we'll be seeing the release of calcium ions from the sarcoplasmic reticulum. So these green spheres are the calcium ions, and they are coming flooding into the sarcomere. This is why we needed the sarcoplasmic reticulum to store calcium, so when it's needed, calcium will be released. So I'm going to stop this animation here. Here is the relaxed muscle. Calcium is on its way. So the myosin head groups in green are separated from the thin filaments, and the troponin molecules are these brown molecules. Tropomyosin is this white twisting molecule, and the actin are the purple spheres. So myosin, to cause a contraction, needs to approach, get closer, and grab onto an actin molecule. In a relaxed muscle, because of the separation, this is not possible. So let's take a look how calcium coming from the sarcoplasmic reticulum will change that. So here are the players, and here comes calcium binding to the troponin. And take a look at what happened with troponin. It changed shape. The tropomyosin also twists around, and this will allow the contact of actin with myosin molecules. So here's the light switch molecule troponin now turned on by calcium, and the binding sites for myosin is now open. The thick filament approach the thin filaments, and now we are seeing the first action of contraction. The myosin head groups are dragging on the thin filaments. So we had sodium ions rushing into the cell, forcing the sarcoplasmic reticulum to release calcium. Calcium came into the sarcomere, bound to the troponin, caused this troponin to obtain this on shape, and this change in the thin filament allowed the actin to be contacted by the myosin. And once that's possible, myosin automatically grabs onto the actin and carries out this movement, dragging the thin filaments towards the middle of the sarcomere. So let me see if I can move back out of here. This process is called the excitation contraction coupling. We started with excitation, flow of sodium ions into the muscle cell, and we ended up with contraction, moving of the myosin head groups, grabbing onto the actin, and dragging, moving the thin filament towards the middle of the sarcomere. So once the myosin head groups are contacting the actin, there is the final set of events that we'll be looking at. This is called a contraction cycle, because for every contraction, think about the frontalis muscle to raise the eyebrows. hundreds of thousands of myosin head groups need to grab onto actin of the thin filament, and sometimes they have to release the actin and grab onto a new one. So maybe this is where this animation is going to be really useful. So we're going to look at a minimal version of this contraction cycle. And I think in a previous lecture, I've shown this animation, but this is good. So think about any muscle like the frontalis or diaphragm or the orbicularis oculi contracting. During that few seconds of contraction, myosin head group needs to grab onto several actins and keep pulling, releasing, bind again, pulling. So this is the contractions. We'll see several contraction cycles here. So this first contraction cycle is partially going. We'll be seeing the power stroke, the actual movement of the myosin head group in a minute. There it is. So this was the power stroke. The myosin head group grabbed onto the actin and dragged the thin filament. But now, if the myosin wants to grab onto this next actin molecule in blue here, the myosin head group has to release the thin filament. This is a part of the contraction cycle. This is the process where we need ATP. So let's take a quick look at this. You'll see a flash of ATP coming into this. There you go. And with the ATP action, myosin released the actin and was removed, detached from the thin filament. And if there's calcium in the system, then this myosin head group is going to grab onto the next actin and pull again, drag the thin filament. Of course, we'll need another ATP to detach the myosin head group. And this goes on hundreds of times during muscle contraction. So this contraction cycle, myosin head group being far away from a thin filament, then contacting the actin, doing a power stroke, and then releasing, those events are called a contraction cycle. So our illustration, I think, in a textbook or on this slide will make a lot more sense after that, I hope. So here are the steps. When there is calcium on the troponin, the myosin head groups will stick to the actin. Here is the binding of the myosin head group to this yellowish actins here. Here is the pulling action. This step 3, the pulling action itself is called a power stroke. But now, if this myosin head group wants to grab onto another actin, the myosin head group needs to detach from a thin filament. And this is what's caused by ATP. So take a look at ATP comes here. ATP binds to the myosin head group and forces the myosin head group to release the thin filament. And also the myosin head group will straighten out and ready for the next binding to the actin and next power stroke. So ATP, the energy, is used in this contraction cycle to detach myosin head group from the thin filament and also make it ready for the next binding action and next power stroke and moving the thin filament. So because hundreds of these power strokes play out during muscle contraction, this will bring up the question, where is all this ATP coming from? Muscle contraction puts a huge demand on ATP production in skeletal muscle cells. So this is a brief summary of this. And here's an overview. So from the delivery of nerve impulse, we have the excitation contraction coupling. And once the sarcomeres are shortening, we need several of these contraction cycles, the binding and release of myosin head group to the actin and the thin filament, then will be moving closer to the middle of the sarcomere. Just a few entertaining true or false questions here. Lecture exam 4, we'll have a huge parts of it coming from chapter 10. So once the muscle is contracting, this is great, but there comes a time when muscle needs to be relaxed. So how do we achieve that? And it's very, very simple. We just stop the flow of nerve impulses to the neuromuscular junction. So when there's no nerve impulses, the acetylcholine will be degraded by an enzyme called acetylcholine esterase. When there is no acetylcholine, the sodium channels will be closed and there will be no sodium flow into the muscle cells, so muscle action potential stops. When there is no sodium flow into the muscle cells, the sarcoplasmic reticulum calcium release stops, these channels close. Now here's an interesting aspect of in the life of a muscle cell. When the muscles are relaxed, the calcium will be vacuumed out from the sarcomeres and pumped back into the sarcoplasmic reticulum. So the sarcoplasmic reticulum just gets ready for the next contraction, loading up on calcium, vacuuming everything out from the middle of the muscle cell. Inside the sarcoplasmic reticulum, we have a protein calsequestrin that acts like a molecular shell. Calsequestrin allows the packing of huge amounts of calcium in a small space, and sarcoplasmic reticulum takes advantage of this calsequestrin protein. So once we remove calcium from the sarcomere, then the troponin will regain its straight shape, its off shape, and the actin is no longer allowed to contact the myosin group. So contraction, contact of myosin with actin is no longer possible. So this is the process of relaxation. The chemicals inside our muscle cell cause an automated sequence to be activated. Just how automatic it is, this is a great example for that. We have situation for the rigor mortis a few hours after death of an individual. And this is a time when skeletal muscles lock up and become completely immovable. Now, why would a skeletal muscle lock up in strong contraction? This person is dead, nerve impulse is completely stopped. So in this 3-4 hours after death, calcium ions start to leak out from the sarcoplasmic reticulum, flow into the sarcomeres, and start a contraction cycle. The myosin head groups bind to actin, and a power stroke starts, but the power stroke comes to a stop. The thick and thin filaments, the myosin and actin, cannot be separated because there's no longer ATP around. ATP production stops once oxygen is no longer delivered for the mitochondria. So the skeletal muscles stay in this locked up rigor mortis state because we cannot separate the thick and thin filaments because we have no ATP here. And this is just an illustration how automatic it is for a contraction cycle to start. All it takes is calcium flooding inside the muscle cells. even in a person's muscle who's no longer alive. I'm not going to say much about this length-tension relationship. All I'm going to say, and when you look at this graph, or we can look at the text version of this, that there's an optimum overlap of thick and thin filaments that's needed for the best muscle action. In a resting muscle, it's 70 to 130% of sarcomere length that is produced. And when the sarcomere length is less than 70% or higher than 130%, muscle strength will be compromised. It would take us a really long time to go into detailed explanation. So the muscle length has limits, whether being too long or too short, to exert optimal power. So there is this Goldilocks muscle length and sarcomere length between 70 to 130% to provide the optimum, the maximal power from this muscle. So let's talk about the last part here in this lecture, ATP. We saw that in the contraction cycle, for every separation of thick and thin filaments, we need ATP. ATP is used up in that process. ATP is split into adenosine diphosphate and phosphate when energy is released. And if you look at the ATP in a sarcoplasm, it can provide enough energy only for a few seconds of muscle contraction. So ATP needs to be replenished. Think about as you're sitting, perhaps watching this recording, you have several dozens of muscles that provide an erect position to your torso. Those muscles need to work for minutes, for hours. So ATP needs to be produced on site. And there are 3 different ways of ATP production that we'll look at. So here is a quick replenishment of ATP, and let's focus on the right side of this image. So here's ATP used up for energy production in muscle contraction, and the energy is released. And here's the ADP, adenosine diphosphate, as a byproduct. We can recharge this ADP quickly into ATP by using creatine phosphate. Creatine phosphate is an organic chemical located in all muscle cells. And when we have enough creatine phosphate, the phosphate group will be added to ADP. And now again, we have a high energy content molecule, ATP, that can be used in muscle contraction. So we have enough creatine phosphate in our muscle cells to replenish ATP for about 15 seconds. This is clearly not enough for those several minutes or hours of maintaining standing or sitting posture. We also have 2 sets of chemical reactions, cellular respiration, that provides energy for longer periods of time. Both of these energy producing processes start with glucose. So let's take a look at anaerobic cellular respiration. So glucose is split into smaller byproducts. In this case, glucose is split into pyruvic acid. This reaction produces ATP, and so this is one way to replenish, produce new ATP in the muscle cells. So this also has limits because we can produce enough ATP for 30 to 40 seconds. Again, if you think about muscles that provide standing or sitting posture, those muscle need ATP much longer than 30 to 40 seconds. And so here is the solution: long-term, large amounts of ATP. Over 90% of our ATP is produced in muscle cells by mitochondria. They are very efficient, and not only they use the pyruvic acid, they can use amino acids, fatty acids, because mitochondria use oxygen. This is not called aerobic cellular respiration. And this is the way ATP is produced for most demands in the body, mitochondria being the most efficient ATP producers. Just how mitochondria can use amino acids, fatty acids, pyruvic acid to produce ATP. The details will come to us in AMP2 in chapter 25. And as a last aspect of this lecture, let's talk about muscle fatigue for a minute. Muscle fatigue is some kind of a chemical deficiency inside our skeletal muscles. This could be a insufficient amount of acetylcholine. This could have a number of reasons for it. So muscle action potential is very, very weak when there's little acetylcholine in the neuromuscular junction. We could have a depletion of creatine phosphate inside our skeletal muscle. We can have a calcium deficiency inside the sarcoplasm. We can have insufficient oxygen or glycogen, or buildup of chemical byproducts, adenosine diphosphate or lactic acid. Now, this is a curious thing, muscle fatigue, because we consciously perceive, we consciously feel that we are tired. So it is a change in the chemicals inside the skeletal muscle that produce this nerve impulse that is going from the muscles to our brain to inform us that we are tired. And again, chemical changes inside the skeletal muscle will be creating this feeling, creating this perception that we are tired. So I'm going to stop here. This is all we want to say in lecture 2. And let me see if I can stop screen sharing here. Right. So this is all we wanted to cover in lecture 2. Because we are meeting on Monday and covering the last part of chapter 10, please bring any questions with you. That's if something is unclear from this second part. There's a lot of very new processes covered in here, new names of chemicals. So, and of course, you don't have to wait till Monday. Send me a message if you have any questions about this part in the life of muscle cells. So I'm going to stop this recording and put this in your class shell very, very soon.